I'm mad at Disney, Disney. They tricked me, had me wishing on a shooting star. But now I'm 20 something. I still know nothing about who I am or what I'm not. So call me a pessimist, but I don't believe in it. Finding a true love kiss is bullshit. Cause I felt sad love, I felt bad love, sometimes happy love turns into giving up.
Hi everyone, how are you? So, in a continuation of the chapter 4, now let's take a look at the genes, the structure, and how they are regulated in their expression. So once again, I emphasize that what I mean by gene expression is an RNA making, so copying of RNA, which is we call there is a specific terms describing this making of RNA in the form of a complementary copy of any particular segment of the DNA. We call this a transcription. So here actually very important definition of the gene can be drawn from this aspect. So overview once first of all let's consider, so what is this gene? So there is some segment of the gene, as we have defined a certain area, not everywhere but certain area of your DNA in the chromosome can act as a gene because they can make the RNA. So for now, minding only the narrow definition of the gene, which is only related with the protein coding information, the messenger RNA. Yes, the messenger RNA will generate any information necessary and required for the protein. So this information somehow is converted into the protein building tasks. So this information is in this aspect, the role of this messenger RNA is actually the messenger, the bellboy. So the information contained in the DNA is somehow temporarily transferred into the form of this messenger RNA, and now this guy is playing a key role of. So this one is able to. It's a one and only. This ribosome is capable of interpreting this information. He can read, or he or she can read the information, what it means. So that's the overall job of this, the transfer of information originally in the DNA into the form of actual tangible protein. So then when we refer to this gene expression, then we can describe it as any kind of activity of RNA making from DNA. And in particular, by the way, this template, the concept of this template. So here, if you have these two upper and bottom, two DNA segments, so either one of them, this top and bottom, let's just put it, this DNA, double strand, if this area somehow, this area act as a gene, then the RNA, depending on which top or bottom. So if this RNA is trying to be synthesized according to the complementary base pair of this top strand, so this is growing RNA chain, like this, it moves. Then this happens to be the template DNA, which is on which RNA is trying to copy, and then this bottom strain is automatically becomes non-template, right? But here you will see some surprising reversal of this fortune, in that This non-template DNA, another name of this non-template DNA is sense DNA. That indicating that making sense. And this template DNA, even though it is serving as a information provider, but this template DNA is another name for this template DNA is nonsense DNA. And you will, if you think about for a minute, then hopefully you can understand why is that so. Because always these three, the template DNA, bottom non-template DNA, and this growing RNA, they have some specific three-way interrelationship in terms of their complementarity. So in other words, if you examine the RNA sequence that is just being made, then you will soon easily realize that, oh, that RNA sequence is it's identical to that of the bottom, the non-template DNA. The only difference is a conversion between uracil and thymine. Instead of the thymine on the DNA, now the uracil, the thymine is not there in RNA. So that's the only trick. So, like I said, this transcription business is usually one of the way you can describe in pictorially is like that. So, and another thing is also in the, which is the same in the case of a DNA synthesis, but in the case of a RNA synthesis, also the direction of this growing chain of messenger RNA, guess what? From where to where in terms of its direction. It's also from 5 to 3 prime and messenger RNA synthesis. And also, when this messenger RNA is trying to pair with its template DNA, okay, so here in this cartoon, this must be template strand. Okay. So its direction, if DNA, the template DNA strand, this goes like this is a 5 prime and this is a 3 prime, and then RNA strand should also migrate this direction from 5 to 3 prime direction. So this is the direction of the RNA being made. So it always has to be this anti-parallel as we have seen. So it goes for all the existing nucleic acid, they have to pair in this way of anti-parallel mode. So this, not all genes are active. So first of all, what's genes? And even if you are entitled to become genes, still it doesn't mean that you always have to be really up, diligent, 24/7, like convenience store. No, you don't have to be. So not all genes are active on a given genome, in a given time or a given location in if you were multicellular organism. So in order to distinguish this difference between this actively transcribing gene versus something that in a status of so to speak, taking a break or on vacation genes. So here goes the active genes versus inactive genes. So in active genes, now we can point that any area which is capable of making RNA, so you do have a potential, right? So you are absolutely, legitimately, you are gene. However, you are not doing your job. Then you are inactive gene. And on the other hand, you are actually, yes, structurally, you have everything to become a gene. Then you are actively making. You are actively engaged in transcription. Then we call those areas as active genes. So on the other hand, this inactive gene, or sometimes it's called the silent genes, they are making virtually no copies or very little copies of RNA. But then this status, as you can easily imagine, it is not something that's fixed. So it is dynamic. So in a given gene, sometimes it can be active, some other times it can be inactive. Also, it also depends on different area of your body if you're a multicellular organism. So the given gene may be active only in a specific time limit, or location, so tissue specific or time dependent. In other words, we call it as a developmental specific genes. It is a very important concept of this differential gene expression. So using this as a trick, so now the key of the being multicellularity, as I mentioned earlier before the midterm exam, the very critical point of being a multicellular organism is it is not about the numbers. It doesn't matter how many cells you have, but those areas, different areas, should be different. Differentiation. So this keyword, the differentiation, which is a hallmark of multicellular organism, is actually achieved by this mechanism, so-called differential gene expression. So in order, before we actually get into to understand the real tricks of achieving this differential gene expression, first then, who are the ones actually involved in the gene control, the gene activity control? Obviously, there should be the real guy, the RNA main character, which is RNA polymerase, which is directly responsible for the synthesis of RNA out of this your DNA template. So this is here, this big thing trying to represent the RNA polymerase. So it can actually open up a specific area of the DNA, and this RNA polymerase one by one insert the temporary. So this is now what you see over here is a temporary complementary base pairing between RNA and DNA, and then this RNA polymerase move down the area. So this should be 5' to 3' direction as far as this messenger RNA concerned. So this is the main character which is responsible and catalyzing this so-called polymerization reaction, right? However, in order for the RNA polymerase to be able to start this catalytic activity of RNA polymerization, it needs some very specific area on the DNA of the gene, right? We call that as a promoter. The area, just like the promoter's role is, a specific area of the DNA sequence. So if we, for example, let's say this bar represents some specific gene area, okay? And actual RNA synthesis start at this point. So if this was the starting point, so the RNA, whether this is messenger RNA or whatever RNA, so this copy from here to the end. If this is the case, so this is a defining unit of actual functional gene, then what you absolutely require for this, so this will be catalyzed by the big RNA polymerase, right? And there is very specific area on this DNA. This now we call as a promoter, to which, so it's always an interaction. The magic, all those wonders of the lives, mostly are the results of a very specific interaction or association or binding, so to speak. We have already, within this short period of this the class, we have already witnessed several different occasions of these interactions. Okay, even the virus entry of this the nasty coronavirus's entry also depends on this specific interaction with this some specific receptor on the cell surface of the specific tissues, just like that. So this is not also an exception in that this RNA polymerase, I will just abbreviate this RNA polymerase as RP. This RNA polymerase protein, if it wants to start this job, then it has to be able to recognize this very specific promoter sequence. So this is some kind of a DNA sequence element, which specifically has to have some very specific DNA sequence, just like as we have seen in a previous lecture that you have some sequence repeat, and also the restriction enzyme, they recognize some very specific DNA sequence motif and then cut it. And so it is one of such extension of those many different examples of this interaction. So this is called the promoter. So any and every gene, if they really want to become gene, which means they are capable of making RNA, then this is the foremost and the absolute, what I do? The absolute requirement of having this very short segment of DNA area having very specific DNA sequence, which we call as a promoter, right? And where are those promoters are on in terms of this, relatively speaking, in terms of the gene, is what we can say is that it's upstream on the coding region. So now we have several different terminology already. I am throwing at you. So in this figure, the coding region, so whole entire any given general unit of a gene consists of two different areas. One is called coding region, and the other one is called non-coding region, and both together make up one functional gene unit. So here in this figure, the coding region. So by the way, the coding region is directly related with the RNA synthesis. So to which that RNA is trying to match. So that the copy of RNA, which is having the complementary relationship, is generated. That area is called coding region. But usually the coding region is usually assigned, reserved for trying to describe the messenger RNA gene, because it is coding. When we usually think about the coding, means it's just like synonymous to the protein coding information. But if we remind ourselves that not all RNAs are messenger RNA, which are the directly protein coding genes, then this coding region should also be applied to the case of non-coding the DNAs, which are making the ribosomal RNA or transfer RNA or some other weird names of RNA, including microRNA and the the small nucleolar RNAs, as we have seen in previous lectures. So anyway, so here in this area would be the coding area, because that the length of the RNA generated from this particular gene will exactly match, at least in the early stage, or only this area only. So that's why this particular area is a coding region, right? And you may actually begin to wonder what are these in the coding area. What you see is some weird names like exon intron. What are they? Yes, we will get to that in a minute. And so this is a coding region. And by the way, this coding region of the gene is that of the messenger RNA, which is related with the protein coding information. And there is another non-coding region, which is not actually present in the RNA. So if you generate RNA, which means that the gene expression, if this gene was expressed, so as a result, RNA is made And on that RNA, any of this non-coding region information is not going to be present. It's like play or movie. So what you see on the screen is just them and actors, okay, actors and actresses. However, in order to generate that movie, behind the screen there are lots more than actually more than number of these actors. There are so many other different steps helping in many different ways. Then, as a consequence, that was possible to have that some specific play or movie can be made. In the same thing here, in order to generate this particular area of the segment or length of RNA, you actually need some more extra area within, or not, I shouldn't say within, but near that gene area. So then we collectively call those areas as non-coding regulatory region. Yeah. So, and yeah, where were we? Well, I was just talking about before this slide. I was talking the upstream, right? Why the promoter? We can usually describe the location of the promoter as like it's upstream of the coding region, and you see. So if this was the coding region of the gene, that means this is the starting point of this transcription, and the promoter is just right before the start of the transcription, because this promoter by definition, this promoter area is the area where the RNA polymerase need to bind. So here the RNA polymerase must come over here, okay, and bind and start. So as a result, you have this, right? However, if you examine the sequence of this produced RNA, in this case, must be a messenger RNA. Nowhere can you find any evidence or traces of the information about promoter of this gene, because promoter is not there. It's not part of this transcription, or the product of the transcription we call as a transcript. The product, yes. So then, by convention, like as you see, it's all, whether it's a DNA or whether it's an RNA, we call that as in terms of this relative direction of whether it's a 5' end here, and this must be the 3' end. So we regard usually 5' end as the head area, and the 3' end as some kind of similar to the tail area. So upstream means before this 5' end, the very first messenger RNA nucleotide, just like before. So that's why we call this area as what? Entire area of this, which is before the 5' end of this starting, the very start point of this transcription. And this must be the upstream. Yes. And how would you call then this down the road area? Yeah, of course, downstream. So this upstream and downstreams are always this relative indication or terminology with respect to this the RNA or gene product generated. So that's I've been just preaching for a couple of minutes. Even though this promoter is not part of this transcribed RNA, but it is essential. Without it, you cannot even start the gene the transcription. So that's why actually we can. So any gene, how do you know whether a certain area can function as a gene or not? Then you can, one clue to distinguish whether those area looks like a gene or not gene is whether that area contains the promoter nearby the starting point of that transcription. So if you have a promoter, yes, then it is a real gene. So with promoter, then those area can function as actual gene. Whether this gene is going to be active or not, it's just a different story. It can be active or it can be silent, depending on the circumstances. But to begin with, whether that certain area of this area looks like being able to code the protein, some specific protein. I know the specific sequence, but still, if you cannot find this promoter nearby, then forget it. That it isn't just garbage, not going to be able to function as a real gene. Actually, so with promoter, this gene without promoter, then cannot function as a real gene, right? So actually, there is a very specific examples of in this incidence. It's actually called pseudogene. In the genome, including human, many some unit of DNA sequence, apparently it look like gene, but it never can be expressed. So like even if that area, in terms of this coding sequence, presumable coding sequence, very much resemble. It's strikingly similar to, for example, hemoglobin gene. However, that area can never be able to express. Why? Because it doesn't have any promoter. And that type of a gene-like element, but not having any promoter, so that cannot be expressed, we call it pseudogene, so false gene. And yeah, there are several areas that we can find the pseudogene area in our genome. So the RNA polymerase, it was the main guy. We absolutely need them. However, is that it? No, answer is no, because in addition to the RNA polymerase, what we also need is many different types of those friends of RNA polymerase. They are all proteins. The ones I have just mentioned, that is protein factors in previous lectures, right? These collectively called transcription factor. Because there are so many different, so I will put S in parentheses. So these transcription factors are proteins that help to stabilize the interaction of the RNA polymerase to the promoter. So for that aspect, yes, these transcription factors really help in the rate of gene expression. So transcription factors greatly enhance the rate. Well, the other way we can describe this is you can enhance the efficiency of gene expression, right? How? As I mentioned just now, by stabilizing the binding of RNA polymerase to the promoter. Because once again, the binding of a promoter is not something like the helicopter is trying to land on the helicopter the platform, but actually it is a really exciting and dangerous 3D incident. It's just like a rope in the 3D space, and this RNA polymerase, poor RNA polymerase, is trying to grab a specific area as its own recognition sequence and binding site as a promoter, and then grab. Then now this acrobatic movement of RNA polymerase to open up and then move through, and in the meantime synthesize this RNA will begin. So this is really not easy and very dangerous to for the sake of this RNA polymerase activity. So to more stably, in order for this RNA polymerase to stick to this particular promoter area more stably would greatly help the speed of this RNA synthesis. That's what we call as a rate of transcription. Right. So this rate of transcription will be greatly increased by the presence of these many different proteins nearby. So those proteins will also try to find its own recognition site, their own niche on nearby area of the gene, right? And then, hey, they will cheer for, like emotionally and also physically, directly, trying to make some kind of a grab with this RNA polymerase. Another very important function of these factors, transcription factors, is, as we have seen briefly in the case of this heterochromatin and euchromatin. So as we saw, some, not all, but some transcription factors can convert certain heterochromatin area into a more opened up euchromatic area. So that then now after that RNA polymerase can now recognize promoter and then land. So this accessibility issue, so some transcription factors can also increase this accessibility of RNA polymerase to the promoter area by providing such valuable trick, which is converting certain heterochromatin area into euchromatin, like this. So the a different figure, but explaining the same thing as we have seen that in the previous lecture. So this transcription factor invite some very specific enzyme, the protein enzyme, so that together they can get rid of some of the roadblock here in terms of nucleosome, so that all this area suddenly become naked, so that opened area. So now this promoter area can be exposed. So then now that the RNA polymerase RP can now find and have a easier access to is. So these two different modes. One, stability, increasing the stability of bound RNA polymerase, and another, in some other cases, this actually transcription factors even make this a pre-clearing of those area, so that this RNA polymerase can have a much better access, so that they can now start this transcription. Right. So practically, even though we all recognize this RNA polymerase as the guy, without it the transcription cannot even start. However, the RNA polymerase itself alone cannot do anything, not a single thing. So if for any genes, that if that gene is going to be expressed at all in a kind of a noticeable level, so some genes can be expressed at very undetectable, like under the radar level, and those gene expression in practically in our cells are totally ignored, that outcome of such gene expression. So if anything, if any gene is to make any difference, if you want to become a difference maker, then you should be able to bring some other actual difference makers. And in here, what I say is that the real difference maker is these transcription factors. Once again, they are all different types of proteins, and all of them are like, they help transcription. So we can revise the, so because of that we can actually revise the definition of this active gene as to the active genes are something that transcription factor, at least one, more the better. So some different, usually the several different multiples of transcription factor proteins interact together in a given gene promoter. So now in this scenario we call that as an active gene. And why certain genes are inactive? And then, oh, sorry, no transcription factor is available at the moment. We'll call you back. Is the situation. So then now we can more easily understand the differences of why a given gene, even though that gene is exactly the same gene, sometimes this gene is active and some other times it's not active. It's not usually determined by the presence or absence or activity of the RNA polymerase itself, but actually rather indirectly by the availability of these transcription factors. If you have, luckily, if you are as a gene, if you are lucky enough at the moment, if you're lucky enough to have a luxury of bringing call your friends, okay, why don't you come over to my place? And then, okay, fine, if some transcription factor protein can come over to you in your nearby area, then you are lucky enough to be able to active in terms of the gene. And some other times, if you are unfortunately cannot invite any others, then you have to put up with your fate as an inactive gene. Okay. And what determines this transcription factor binding? Can a specific given transcription factor, without any discrimination, can bind any different genes? We do, mind you, we do have more than 20,000 different genes in our genome. So then all these transcription factors are so busy in like substituting all these different areas of the promoter? No, that is not the case. Each unique gene carries some very specific, I'm here for you only, like very specificity in terms of the DNA sequence. So it's all these different transcription factors interact with a certain area on the promoter by this sequence specific manner. Okay, so this sequence specificity is maybe it's just even too much detail for those who are not really involved in this business. But just in case you are wondering, let's say the big block is a coding area of the gene, coding region, and then let's say this, so that there must be just right upstream of this coding region, there's going to be a promoter, right? And usually this promoter sequence is more general. So in other words, RNA polymerase, the same RNA polymerase can recognize the promoter sequence equally as well as the promoter sequence present in many other different genes. Okay. But the difference is this: some of the DNA sequences present like A, B, some C, several different DNA sequences. All these different DNA sequences are specific for the transcription factor binding site of different proteins. And these different DNA sequence, which is for the transcription factor, we call as what? Enhancers. So these enhancers are, once again, the DNA sequence, just like the promoter is the DNA sequence, but they are more specific. So the variation is higher depending on different by the genes. So in that way, each given unique gene usually bring different type of protein as their transcription factors. That's how usually it is being managed. Okay, so this enhancer, And if there were angels, then there is a devil. So uh, not only usually gene have this enhancer sequence, but a lot of times this gene also has some different type of DNA sequence element, like the interfering sequence. And those interfering DNA sequences are like a binding site for the bad guys, negative guys, the proteins. So all those transcription factors, there are some of the transcription factors actually acting as an inhibitor. So this some proteins inhibit transcription. It's always like a tug of the war. Is if some friends will help you and some other like enemies will try to stop you. It's always like a balance of power is going on for any given gene, and those proteins are called repressors. So once again, to make this thing clear, like here the transcription factors is a protein, and counterpart of the transcription factor is the repressors. Actually, some people distinguish a little bit differently. Some is between transcription activator and transcription repressors. But so you know what I mean. So there is some kind of negative guy called a repressor specifically. When you usually refer transcription factor, then usually it means as a positive guy. So these are all proteins. Then for each of this protein, that corresponding DNA element to which this these two different kinds of protein factors specifically bind. Then here for the transcription factor, there is an enhancer. So DNA sequence element. For this repressors, what do you have? This is a silencer. So these two different types of DNA sequence element, enhancer and silencers, are all DNA sequence, or some other times it's called DNA element. Is what they are called. So the silence versus enhancer, and transcription factor versus a repressor. And so this, the pink box, the marked with the pink box, the entire area is actually one functional unit of the gene. However, now we can distinguish these two area as one gene unit, but you have a coding region plus the regulatory elements. So here the coding region should be this area only, right? Which is equal in length to the produced RNA. So that's the coding region. And the rest of the, within this box, within this pink box, the rest of the area would be so-called this regulatory region or regulatory element or control element. So here in this figure, what we can recognize is either in the 5' upstream or 3' downstream, the regulatory element can be present, is what we can, just by looking at this figure, that's something that at least we can draw the conclusion, if that was the case, right? Well, actually, that is some kind of the conclusion that I hope that you can make instead of what's the point of trying to insert any very detailed trivial piece of information into your already poor soul or poor brain But just by looking at this, given the information of like this, oh, here are how things are like that, then by looking at, then you can, oh, okay, so if then it must be this or that kind of uh, observation based upon the proper information, then you arrange and understand that information, and then you can draw the corresponding the conclusion or accordingly is actually the most important aspect of this level learning, I guess. So here in this, the smaller pink box should be what the coding area, coding region of any given gene. And if this was the messenger RNA now, if this was the messenger RNA of eukaryote, the prokaryote don't have anything like this. So what is this exons and introns? Some very confusing terminology. But if you have to deal with this eukaryotic messenger RNA gene, is especially, then your coding region usually almost always, but not always, usually consist of exons and introns. And here on this figure, exon and intron on the DNA is marked with different colors. One exon is colored in stronger blue color, and intron is in the fade blue colors, like here. And they are transcribed, both of them. In this figure, what you see is, okay, fine. So regardless of whether you are part of the exon or part of the intron on initially in the coding area of the gene, both of them will be present in this, the product of this transcription, as you can see. Right, that is true. So on the RNA, this RNA, by the way, is called primary RNA transcript, as if there is a secondary RNA transcript. Yes, they are. So this RNA, the point here is eukaryotic messenger RNA will undergo some editing, trimming process, unlike any regular prokaryotic messenger RNA. So this is one such more complexity aspect added on to the eukaryote as after they become more complex organization compared with the prokaryote. So this is in primary transcript, which is before going through the actual final trimming or processing steps. In the primary RNA transcript, yes, both exon and introns are present. And you compare the actual product, the finalized product here in this slide as a final mature messenger RNA. Anything you have noticed that something that missing in the final version of the messenger RNA after this processing, RNA process? It's called, collectively, it's called, entire task is called RNA processing. Right. So what you can obviously see is what who are absent in the final version of the messenger RNA. Obviously introns. All those area initially labeled as introns are all gone here. And only, so now only those exon, the area originally labeled or recognized as the exon block, are now combined all together. Okay. So how would you describe this process to generate this, the final version of messenger RNA, starting from this primary RNA transcript? So this entire process is called RNA processing. But one obvious thing you can notice of many different types of RNA processing, what you can see over here is the introns are all removed. Yes, and exon, all those exons are put together. The term describing such putting together task is called splicing, RNA splicing. So exon splicing, intron removal and exon splicing is the one of the major step of the overall RNA processing of the eukaryotic messenger RNA, as you can see over here. See, so as a result, all those introns are removed. So that's the coding region, and here we have just briefly defined the exon and intron, which we are going to finalize in a minute. And the promoter area binding site for the RNA polymerase is obviously not a part of this final version of the messenger RNA. And looking at this control element a little more, and these enhancers in the upstream, those enhancers binding site for these regulatory proteins, and those are control element of the 5' upstream, and also you can have the downstream control element too. So this is the regulatory region of the gene that contain some of the important control elements, namely examples are like enhancers and silencers and promoter. Those are the DNA elements, right? Each of which is used for different interaction with different proteins, as we went over, right? And so this promoter, I just try to make a silly cartoon of on what types of different protein factors can somehow influence the stability of RNA polymerase. And we may actually, you may still wonder, okay, I can understand some of these guys because they are physically very present in a very near the close proximity to the promoter. So yeah, I can understand those. What are your jobs? What are you doing? What's your contribution in this gene expression? Well, one thing, at least for one thing that I just mentioned, that some of them actually can clear all those blocks of nucleosome so that this RNA polymerase can have a better access. And also, in other cases, these guys can have real physical interaction influence from such a long distance somehow, just like making some of the work. We'll see later in this lecture. And sometimes life's not always perfect, so sometimes these kind of bad guys may be able to bind in between, so they try to interfere all this kind of a productive job of gene expressions. That happens, and then in that case, then you should be able to come up with a way of getting rid of this guy. How can we? And that's always a kind of a challenge for any given gene to be able to properly express. Okay, so now getting back to this, the mature RNA business, so that now as we can compare easily this primary transcript versus the final mature messenger RNA, then what we can define for an exon is perhaps this. Exons are segment of messenger RNA coding region. Yes, so only coding region there are like whether you are an exon, the part of an exon, or a part of an intron applies. If you are in the controlling element, that doesn't apply. Okay, that's irrelevant. So exons are segment of the messenger RNA coding region that still can be found in the finalized version of messenger RNA, and they are all exons. Why they are important? Because they are finally the ones that can make some kind of a say, have say to what kind of proteins are going to be made. Imagine if you were an intern. Maybe initially you may actually daydream so that I can make some difference because my DNA sequence will be able to have some kind of control in what type of protein is going to be made. And sorry, that is not the case because you are going to be eliminated, so you don't have any influence. But exons are different, so they are the one that can have some kind of specific influence in what kind of proteins are going to be made out of this particular gene. On the other hand, the intron is this segment of the messenger RNA coding region that is removed from mature RNA. Right. So that's the difference, a structural aspect of the difference between the coding region and the control element. And in particular, if you are in a coding region of a messenger RNA of a eukaryote, you do usually have this intron and exons. And let's just put up the more important question, seemingly, that why introns are there in the first place. If you're not going to contribute anything in actual final protein make business, then why are you there still? The evolution, the process of rigorous evolution over such a long time should already have been able to find you as an obsolete and then gotten rid of you. But that is not the case. We don't know why. But anyway, let's just first take a look at this. Now I believe the enough background orientation has been given to be able to understand the mechanism of this differential gene expression. So getting back to that original fundamental question of why for a certain gene, sometimes that certain gene is active and some other times is silent, and more importantly that to become a basis of this important feature of being multicellular organism. So the key is the transcription factors, right? As I said, so RNA polymerase itself doesn't really have any kind of the real control power. It's all by the presence or absence of these transcription factors, or how whether they are active, even if they are present, if they are present in somehow inactive states, then they are. Their role is just simply not there. So this availability of this activity, the transcription factor activity, is very crucial in whether determining whether a certain gene is going to be active or not. So, but then that activity is different under certain different circumstances. So that now we have such a thing like some genes are regulated by tissue-specific manner. Then we call it as a tissue-specific gene expression. Very good example of tissue-specific expression. I'm such a dumb enough so that I'm only able to come up with always a one single example of hemoglobin. Always stupid. This hemoglobin gene expression is a very good example of such a tissue-specific gene expression in human and many other the vertebrate animal, breastfeeding animal too, mammals. So that this hemoglobin genes, the function is to carry the oxygen transport function, and the only place you can find the hemoglobin protein expressed, made, is where? The red blood cell. Okay. So then inside of the red blood cell, hemoglobin proteins are abundant so that each hemoglobin protein can pick up oxygen molecule, and while this red blood cell travels through all the entire body, through your blood vessel, that's how you can get the ample, adequate supply of an oxygen from outside. That's how it goes. But then does that mean this hemoglobin gene that is responsible for making this hemoglobin protein Do you think that hemoglobin gene is only present in the red blood cell? The answer is no. If you look at any of the different type of cells in your body, you can always find the same copy of the hemoglobin gene that you can find in the red blood cells. And what is the difference? Why only in the red blood cell that hemoglobin gene is active and then make the RNA, and as a consequence the protein is made, whereas in the rest of your other cells in other tissues, that hemoglobin gene is acting as a sleeping beauty. It's because of this tissue-specific gene expression characteristic. Only that hemoglobin is active in the red blood cell. And this is not the only example. So many different genes behave as this tissue-specific expression pattern, so that only certain different area that gene is active. And another way of modulating this gene expression is sometimes some other genes are regulated here, like in this developmental specific. Examples are, well, in your very early developmental stage, when you're a baby, certain genes are active, and then as you grow older, that gene become inactive. We are going to see actual real life example of such gene later on this chapter. So we then call it a developmental specific gene expression. In both these examples, the key regulating factors are the availability of this transcriptional factor, that they're active transcription factor. So in tissue-specific gene expression pattern of the hemoglobin gene expression, the reason is probably that transcription factor enabling the active expression of the hemoglobin gene is only available in the red blood cell, no other place. And in the case of this, the developmentally specific gene expression, that other types of such transcription factors required for the gene activity is only available in a certain timing in your lifespan. Okay. So here is one such illustration of tissue-specific gene expression in two different types of tissue. One is a liver cell, and the other one is a lens cell in your eyes. So the structure of the two genes, we are going to take a look at these two different genes. One is the albumin, is a protein, a gene making a protein abundant in your bloodstream. Actually, the albumin protein itself is made in the liver, and then they are transported out to travel. It's not travel as in, I should say, as petrol. So they are abundantly present in your blood, and then they have a very important functions. On the other hand, these crystalline proteins are only present in your lens cell of your eye tissues. So then if you look at the gene structure of these both genes, the promoter, the area of the promoter coded in same color, probably you can guess that, oh, this promoter might be recognized by the same old single type of RNA polymerase, and then handled by. What's different is their enhancer area. For say albumin gene and the crystalline gene is different. So then what we can infer here is, okay, perhaps different types of proteins may be able to act on each of these different enhancer area of the respective gene, and probably what you can further anticipate is probably the availability of this particular partneric transcription factor proteins might be different in these two different tissues. So here the first point is both genes, albumin and crystalline genes, are equally present in liver as well as in the lens cell of the eyes. But still, the albumin protein is only made in the liver, and the crystalline protein is not made in the liver. On the other hand, the crystalline protein is only made in the lens cell, but albumin protein in the lens cell is not at all made. Okay, so now we can see the respective difference in the gene activity, right? And what's the reason is the, so this is about the transcription factors availability. Okay. So hope you can recognize this, the enhancer area. Each of these enhancer area can be bound by all these different transcription factors. Okay, and what they do after binding, what you see is here. What you don't see is out of this the frame, some of the looped out DNA. So from the distance, the long distance, even though this enhancer area element is present far upstream at a longer distance from this transcription start site over here. Okay, here can you point DNA polymerase? Yes, this must DNA polymerase. Okay, and the promoter. See, but still, what they can exert the positive influence on this is by having this the intermittent area of the DNA by looping out. They can have this physically intimate interactions between the transcription factors and the RNA polymerase. Okay, so then now you have a gene expression. And what gene is it? The albumin. Okay, good. Then what about here? You also have this different, although different, but some enhancer element present in this liver cell. Okay, you have a liver cell, and now since this is a liver cell, the albumin gene expressed and the crystalline gene is not expressed. Why? Even though you have this enhancer element, but the transcription factors, there's no transcription factors that can recognize and bind and then provide this type of a similar influence and stability for the albumin gene. You don't have it. So sorry, tough luck, you cannot. Crystalline gene is not going to be expressed in this way. However, things are completely reversed in the case of a lens cell, in that now what you have in a lens cell, this area is not occupied by any of these transcription factors, unlike. So you make the comparison. You don't have these guys in this lens cell. Although in this cartoon, this transcription factors appears to be exactly the same transcription factor with this. No, that's not. Actually, that is not the case. So the proteins that recognize and bind to this, the crystalline genes, these enhancer areas, these transcription factors, are the ones that are different. Basically, maybe some of them maybe actually kind of share the core element, but overall they are totally different than this transcription factor group of the albumin gene expression activation. So anyway, these guys are available in the lens cell, but not available in the albumin in the lens cell. So that makes a difference. And as a consequence, in the liver cell, only the albumin gene, as a result of albumin gene expression activity, albumin protein is made, and the vice versa. In the lens cell, only the crystalline protein is being made. And that's why and how this so-called differential gene expression is maintained. And you can apply the same logic to express the difference in timely manner, the developmental specific gene expression. This type of differential availability of transcription factors can be displayed along the different timing. Then now you call that as developmental specific gene expression. Okay, I will stop at this moment and then continue on the next task of this RNA processing for the intron removal in the next session.